Thank you for coming and joining in for our Research Connect series. My name is Chloe Wong-Mercero. I'm a CDTRP research trainee, and I'll be facilitating today's webinar. So I think we're just about ready to get started. A bit about me: I completed my master's in medical anthropology at the University of Toronto, where I now work as a qualitative research assistant. I coordinate storytelling workshops with liver transplant recipients about their experience through the transplant journey. But before we get started, I'd like to take a moment to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota, Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many other Indigenous histories, languages, and cultures that continue to influence our vibrant community. So I encourage you to write your own land acknowledgement in the chat box and introduce yourselves. I'd also like to acknowledge Takeda and AstraZeneca, without whose generous support we could not host these sessions. So the CDTRP also relies on the help of our community. Your gifts ensure we can focus on delivering our mission of supporting excellence in donation and transplantation research. So there'll be a link in the chat box for those who are interested and to donate to the CDTRP. But before we dive into the main content, I'll pass it over to Erica, one of the CDTRP coordinators, for some general updates.
Hi, thank you very much, Chloe. I have two save-the-date announcements today. The first is for our PFD forum. This year's Patient, Family, and Donor Research Forum will be held virtually on June 8th and 9th. Registration is free and it can be done through our website. The link to register will be shared in the chat, and I recommend you go check out that link because you're going to see our framework for our excellent presentations. The second point is for our annual scientific meeting. This year's ASM will be held in a hybrid format in Montreal, with the in-person component in Montreal, Quebec, on December 5th and 7th. We're currently accepting program submissions. So if you have an idea for a session, you have an upcoming project that you want to discuss, maybe you have an idea for a collaborative project that you want to recruit PI trainees on, a workshop idea, maybe a really fantastic idea for debate, something we haven't even thought about yet, submit your ideas. Speaking of submitting, this year there is an oral competition for trainees. Winners from each of the 5 themes will receive a cash prize and free registration. Finally, this year the education platform will be hosting a pre-peer review workshop to help trainees and researchers perfect their manuscripts before publication. All information, including submission links, can be found on our website, and I will post all the links in the chat. That is it. Back to you.
All right. So before I introduce the speakers, just some housekeeping notes. Today's session is recorded and will be posted on the CDTRP website and the YouTube channel. During the presentation, please keep your mics off. And if you have any questions or comments, you can raise your hand with the raise hand button or type them in the comment section. So now I'm pleased to introduce our presenter, Dr. Golnaz Karoubi. Dr. Golnaz Karoubi is an assistant scientist at the Toronto General Hospital Research Institute and the principal investigator in the Latner Thoracic Research Lab. She holds an assistant professor appointment in the Laboratory Medicine and Pathobiology Department and a cross appointment in the Department of Mechanical and Industrial Engineering at the University of Toronto. Dr. Karoubi received her PhD in Applied Science and Engineering at the University of Toronto and joined the Lung Regenerative Medicine Program in the Department of Clinical Research in Berne University, Switzerland, for a post-doctoral research fellowship. She stayed on as a group leader in 2008 to direct the basic and transitional science as related to cancer stem cell and lung regenerative medicine in the Department of Biomedical Research at the University of Berne until 2012. In early 2012, she joined the team of Dr. Tom Waddell at the Toronto General Hospital Research Institute as a senior scientific associate. She was appointed assistant scientist at the Toronto General Hospital Research Institute with the University Health Network in November 2019, and assistant professor in the Department of Laboratory Medicine and Pathobiology in July 2020. Her research interests include the development of practical approaches for the treatment of lung and airway disease. Studies focus on lung, tracheal, and airway regeneration, the use of pluripotent derived airway epithelial cells for tissue engineering applications, and cell-based therapeutic approaches for end-stage lung disease. We are all very excited to have you join us today, and I'll hand it over to you.
Thank you very much for the kind introduction, and thank you for the invitation to speak. So I will just share my screen and go from there. Can everyone see my screen?
Yes.
Yeah, okay, excellent. So I wanted to just kind of give you an update today on what we've been doing so far on our progress towards repopulating long segment tracheal allografts for transplantation. So the lab focuses a lot on airway regeneration, and that it includes the lung as well. But where we're really making significant impact, I think, is in the trachea. And so I wanted to show some of the highlights of that work. So let me just, I think I went too fast. Here we go. Okay, so long segment airway malignancy or trauma and stenosis lead to substantial morbidity and mortality. And when the damage or the site of injury is less than 50% in adults or less than 30% in the pediatric population, this can typically be addressed using surgical manipulation. And so by resection and anastomosis of the injured site, you can get a functional tracheal graft. However, if the injury is greater than 50% in adults or greater than 30% in children, then this has to be replaced. And tracheal transplantation is obviously a practical solution. However, for the graft to be viable, it has to have a functional mucosal lining, it has to have structural support, and it has to have vascular supply. There has been a lot of different products that have been used as a source for tracheal replacements. These include stents, for example, silicone, metallic or bioabsorbable stents, or synthetic prosthesis and scaffolds. These could be solid, for example, solid silicone or Teflon, as well as porous, so various different meshes that are supported to prevent air leakage with, for example, foreign material or sealed with tissues. They can be non-viable tissue, such as fixed, frozen, or lyophilized cadaveric tissue. And they can also be autologous tissues that is available in the form of, for example, free grafts, such as the fascia, dermografts, omentum, vascularized flaps, as well as materials, autologous materials used for tube reconstruction. For example, tube pedicles, which combine skin grafts plus ribbon or costal cartilage. However, none of these have been particularly successful. There's been various different types of limitations, which each of these, some have advantages, some have disadvantages, but for the most part, most fail due to either inadequate revascularization, immune rejection, or epithelial dysfunction, essentially leading to transplant failure. And one of the things that we've been working on is epithelial dysfunction, which I will show you in a bit. And so as an alternative solution, the use of bioengineered grafts for tracheal transplantation has received a lot of attention. And these can include synthetic scaffolds, for example, 3D printed polycracolactin as an example, but there's several other materials that can be 3D printed, or biological scaffolds, and mostly including decellularized tissues and organ systems. And so this is something that we've been interested in, the use of repopulated biological scaffolds. And here, represented here in this schematic is the lung, but really the liver, heart, kidney, various different organ systems. And the idea is to procure or harvest donor tissue, for example, the lung here or the trachea, to remove all of the cellular material, leaving behind an acellular scaffold. And usually this is done through some sort of enzymatic detergent-based system where all the cellular material is removed. And this leaves behind this intact, this structurally intact scaffold that is free of any donor material. This is then recellularized or repopulated with various different cell sources. These could be recipient-derived autologous sources. They can be stem cell populations, and they can, and recently there's a lot of interest in using non-immunogenic universal cell lines to repopulate various different tissues and organs. And the idea is then to use a bioreactor system where the tissue engineered graft is matured and then to implant into the recipient. So that's the concept. And that's what we started doing in the trachea. And this is work by Dr. Siba Hekel, who's a very close research partner of mine. And initially we started looking at fully decellularized tracheal graft. And so what Dr. Hekel did is take the donor trachea, porcine trachea, removed all of the cellular material, and then repopulated this donor trachea scaffold using mesenchymal stromal cells in lieu of cartilage and tracheal epithelial cells from primary tracheal epithelial cells from porcine in order to recreate a graft. She then matured these cells in a bioreactor system for 3 days and then did an orthotopic transplantation into the recipient, following them up for 3 to 5 days to see how they would do. And what we learned from these results was that the use of primary epithelials was very unreliable because of the high heterogeneity and differentiation capacity across the donors. And I can show you some of that work that we've recently found with these cells. And they're unable to yield a large number of cells. These primary cells are typically have slow growth rates, they have restricted expansion, they're not easy to work with ex vivo. And so they're not an ideal population of cells to then repopulate the trachea with. We also found that the reepithelialization process needs quite a bit of optimizing. The 72 hours that she started with were really not enough to produce mature epithelium, and that's understandable. And there was no air-liquid interface in order to mature the epithelium as well. And so the reepithelialization was key. And we also found that fully decellularizing the tracheal graft really resulted in cartilage degradation and in the end, failure of the graft. So these three big limitations we felt really had to be overcome in order to produce a bioengineered graft that could be used for eventual transplantation for these long segment grafts. So then for the past several years, we've been asking two different questions. One, do we need full decellularization? And the idea with that is perhaps all we need is partial decellularization to remove the tracheal epithelium, which is the predominantly immunogenic component, and then we can keep the chondrocytes alive and maintain the structural integrity of the graft. And so there would really be no need to then reconstitute all of the necessary chondrocytes. And then the second question we've been asking is, can we use a pluripotent cell source? So for example, can we use induced pluripotent derived airway progenitor cells that can be recipient derived and in the future non-immunogenic? So I'll begin by showing you some of the work we've done in terms of our partial decellularization approach. And really the rationale was, as I mentioned, the predominant immunogenic component is a result of the epithelium. There's been several sort of larger animal studies and some clinical studies showing that perhaps the cartilage is immune privileged. And that's not to say that it is not recognized. That's just to say that it is not as immunogenic as the epithelium and perhaps will not, can be managed with immune suppression and won't lead to rejection. And so we developed a very relevant short partial decellularization process, and we use a harsh detergent, sodium dodecyl sulfate, at a relatively high percentage, 1%. This was a very short treatment in a double perfusion bioreactor. The schematic is shown here, but if you can imagine that the trachea is within this bioreactor, then there's two different chambers. So in the tracheal lumen is perfused with sodium dodecyl sulfate and then washed with water, neutralized with Triton, and then again goes through a couple of other washes. And then the outside of the trachea, where the cartilage is preserved in a different perfusion system. And so the sodium dodecyl sulfate does not touch the cartilage from the outer part of the trachea. So this results in a very rapid Deepithelialization, it's very effective. You can see here with the scanning electron microscope images that in the native, you see the cells, you sort of see cilia, and then the deepithelialized graft, none of the epithelium is there. However, you can see morphologically from the SEM images that, that the cartilage remains for the most part unaffected. When we look more closely at the chondrocytes within the cartilage, this is just live dead imaging showing viability of chondrocytes in green, and the percentage of compromised chondrocytes that are no longer alive in red. And you can see that in the native, the majority is alive. And in the deepithelialized protocol, we usually get anywhere from 65 to 80% of the chondrocytes being alive. Here is a representative image. You can see that for the most part, they're alive. There is a layer of chondrocytes that is closer to the lumen of the trachea that results in chondrocyte cell death. And we think that that's for the most part because no matter how hard you try to remove the SDS does somewhat permeate through into the cartilage and results in a bit of cell loss. We followed these graphs for over 2 weeks, both in the bioreactor system, and we found that the viability is maintained, and we can maintain the majority of the chondrocytes alive. We also did some characterization of these deepithelialized grafts and compared them to native trachea. They maintained the glycoaminogalactic acids. You can see also maybe with the histology here, you can see sort of like a nice layer of the pseudostratified epithelium, which disappears in the deepithelialized grafts. The gags stained here with Alcium blue are maintained, both in the deepithelialized close to the native. There's no differences in terms of elastin and collagen contact. And then we did some detailed work as well for the cartilage regions. As you can see, no major changes. And then when we did some more specific immunostaining for the basement membrane to look at collagen 4 and laminin, we found that you can maintain the collagen 4 and laminin expression in the basement membrane that has been devoid of the epithelial cells. We also did quantification of the gags in the cartilage and the submucosa, and there were no significant differences as compared to the native tissue. And so we decided to move forward with this protocol, keeping intact the majority of the chondrocytes. And we use our double perfusion bioreactor system, this time modified for repopulation, so for longer term in culture. And we added a few things such as peristaltic pumps, flow meters, thermometers, automated certain flows in order to be able to maintain the bioreactor culture for a prolonged period of time. We did some initial studies with Bs2B cells, which are bronchoepithelial cells. There are a cell line, and we found that we can, the cells attach very nicely to this deepithelialized graft. Presto Blue assay showing metabolic cell activity shows that the cells are metabolically active. You can see here that there's a lag as the cells are attaching and adapting to the new environment, but they grow quite nicely and plateau around a week. And you can see here, again, with the live dead staining, that there's a nice monolayer of green cells that are alive and do well. So this was great for the cell line. The Bs2B cells grew really well. However, when we moved to working with the primary epithelial cells, we could not receive successful reepithelialization. And really, we sort of went back and thought about how can we better augment the reepithelialization process so that it is suitable for primary epithelial cells and for pluripotent ulcers that we would be using in the future. So to do this, we had sort of two different approaches. One was a collaboration with the mechanical engineering department, Dr. Christina Mon at the University of Toronto, really looking at computational fluid dynamics and in silico guided approaches to see if we can enhance the bioreactor design and the recellularization process in order to recreate or create better conditions within the bioreactor system during the recellularization process, resulting in better recellularization. And then the other approach was, can we use a more biomaterials-based approach? Can we find a biomaterial to support epithelial cell growth and differentiation, with the idea that then this biomaterial can be scaled up and we can use it to line the inner lumen of the trachea and transplant a fully differentiated epithelial sheet within the tracheal lumen. So I can show you some of the work that we did with Dr. Aman and her team. We started by looking at the bioreactor design, and we optimized things like the inlet of the bioreactor where air and liquid would flow through, optimized flow meters, thermosensors, automated certain pieces of the protocol. And then we also looked at modeling the actual recellularization process and looking at optimizing important parameters such as fluid flow rate, shear stress, and particle deposition in order to guide the biological experiments. And here's a schematic of the bioreactor. It is a double perfusion bioreactor, as I mentioned. The important thing that I want to highlight here is that we had redesigned the inner chamber. So the inlet is really important. And so we had two different designs that were suggested by the Aman group for us to evaluate. One was a single inlet where you can imagine both air and liquid being flown through as the epithelial cells need the air in order to mature. And then a double inlet condition where air could be flown through one inlet and liquid could be flown in through another inlet. And so we did quite a bit of modeling together to see which of these would be better in terms of both flow rate, shear stress, particle deposition. And so this was the work of Alba Marin and Hank Yu Lee. And they really looked at modeling luminal wall shear stress as well as particle deposition, and with the double inlet conditions and the single inlet conditions. And what I should mention also that within the bioreactor system, this trachea is under constant rotation at various different rotation per minute rates. And so what we were predominantly working with before was somewhere around 5 to 15 RPM. And one of the hypothesis was that perhaps the primary epithelial cells and the progenitor populations may need much lower shear conditions in order to survive and be happier. And so we looked at anywhere from 0 rotation to 15 in our in silico models, and the results showed that the higher you go in the rotation of the trachea, your shear stress patterns are significantly different, and you get quite a bit of quite high shear conditions near the inlet and the outlet. And that will significantly impact the deposition of the particles. So for instance, you can see here in the 15 RPM that because of the high shear, you're getting very sort of heterogeneous deposition of the cells with not much in the inlet conditions, for example, compared to like the outlet conditions. And so to summarize, these results suggested that perhaps lower shear conditions would be better. So then we experimentally validated these different conditions and looked at 1 RPM and the 5 RPM that we had been previously working with, which is the lower of the shear stress conditions. And we found that the 1 RPM condition resulted in better deposition of the cells, both along the length of the trachea as well as along the circumference of the trachea. Whereas with the 5 RPM, you had relatively low cell deposition in the half of the trachea, and then the outlet of the trachea, the outer half, was quite populated. And so we decided to move forward with the 1 RPM conditions. I should also mention that we did a series of studies looking at different flow rates and actually found that the flow rate was not significant in affecting the cell deposition. So we used a 1 RPM and 5 RPM just as a control for longer term biological experiments to see how much impact using a lower shear condition would have in the single inlet condition, which proved to be better. And we found that after 2 hours, the number of cells that attached to the epithelial, to the deepithelialized graft were significantly higher. And at 24 hours, we had a significantly higher number of cells that were alive. We then looked at a longer term, over 5 days in the bioreactor system, and we found that the 1 RPM condition really resulted in nice metabolic activity, a homogeneous distribution of cells throughout the tracheal graft, and we could see a nice monolayer of cells. Now, these were with the cell lines, and so we decided to test these new parameters with the primary epithelial cells and the progenitor cells. And so at this point, we sort of had to decide what is a clinically relevant cell type to work with. And one of the reasons that we decided to move forward with iPS cells rather than airway progenitor cells, that primary progenitor cells, was because of the vast differences in their growth and differentiation. And so there, we looked across 70 different samples, and shown here are a representative 6, which we carried out through 21 days of differentiation to check for the differentiation quality. But sometimes you can't get cells to grow, some of them don't form monolayers, and some have very, very different capacity in differentiation. For example, you can see examples of cells different. These are all different samples where you can see no differentiation, and some with a different morphology in terms of the barrier function represented with the ZO1 staining in yellow. You can see that some have sort of the rare ciliated cell at day 14, some have a lot of ciliated cells at day 14, and then again at day 21, some have none, some have few, some have very good ciliation capacity. And so it is not a consistently reproducible differentiation state that we could work with. And so we decided to move forward with induced pluripotent stem cells. As you know, induced pluripotent stem cells are a very potentially clinically relevant population. They can be derived from the recipient. They can give rise to a number of different cell types. There's no need for embryos, and that increases their use and limits their sort of the issues that there are with ethics. It's not invasive, and so they can be derived from fibroblasts from the skin, for example. So we decided to use these cells on our long segment tracheal grafts. This is a collaboration with Finn Hawkins and Daryl Cotton in Boston, and they're really the leading experts in terms of deriving airway cell types from induced pluripotent stem cells. And very briefly, essentially that we do directed differentiation of iPS cells, mimicking development. And so this is quite a lengthy procedure. It's a 73-day process, starting with making definitive endoderm, then making anterior foregut endoderm, which gives rise to lung progenitor cells. And then these lung progenitor cells are cultured in matrigel conditions as self-assembling organoids, giving rise to airway progenitor cells. This is the point where we use them in the bioreactor system. But in vitro, outside of the bioreactor, if these cells are then grown in trans wells where they're exposed to air-liquid interface culture, they can mature or we can derive mature epithelial, such as ciliated cells or goblet cells, shown here in red for ciliated tubulin for ciliated cells, as well as mucin 5AC expression for goblet cells. You can derive these mature aware cells within 21 to 35 days of exposing them to air-liquid interface. And so we put these airway progenitor cells onto the trach grafts with the rationale and hypothesis that perhaps the more biomimetic bioreactor culture will induce differentiation faster, and maybe we can even get these cell types at an earlier time point. And so we took the airway progenitor cells, which are enriched in P63 and KRT5 expression, and we put them onto not the air-liquid interface, in submerged culture first, so that they can form a monolayer. We found that they nicely form a monolayer over the 3 days. The majority of the cells are alive, and so we were very, very excited about this. And then when you maintain them for another, so at 3 days, they lose the expression of P63, they start to express KRT5 and KRT8 together, and this represents a transitional state in the differentiation towards mature alveolar epithelia. And then when, as I mentioned, we need to have air-liquid interface to mature these cells. So we had to modify the bioreactor system in order to provide air-liquid interface. And you can see in the top panel here, so typically what is done during the submerged culture is just media is flown through the tracheal lumen and then, of course, outside for the cartilage as well. But media is flown through the tracheal lumen and everything's under submerged conditions. And you can see that if you take a native trachea and maintain it in the bioreactor, just in submerged conditions, you're going to lose the mature alveolar epithelium. So you lose the ciliated cells in the epithelial layer. And that's shown here. This is a native porcine trachea that we use to establish the air-liquid contra interface. Now, if we want to expose the cells to air, we tried several different ways, and the best most effective way of providing air exposure in the culture was to bubble air through the liquid at an incline, so that the entirety of the tracheal lumen is being exposed to air intermittently. And you can see here that using this approach, we can maintain the ciliated cells. They are, you can see the pseudostratified layer of the epithelium is maintained. And so we were happy to move forward with our air-liquid interface culture within the bioreactor system. So at day 3, we put the bioreactor at an incline, bubbled air to create the air-liquid interface, and found that we're starting to get a pseudostratified epithelium, as you can see here by the H&E staining, and we already see the presence of goblet cells. And this is basically a mucus-producing cell population in the trachea. We don't see ciliated cells, but that wasn't expected, as it takes a bit longer for ciliation to occur. In vitro is anywhere between 21 and 35 days, although in certain cases where, for example, the primary epithelium was really good, you could see them appearing at day 14. Day 14, you see a nice pseudostratified epithelium. The goblet cells are still there. You see sort of like the rare punctate cilia coming up, but it's not really a fully ciliated layer. And so we're currently trying to optimize the bioreactor settings to be able to do further characterization of these IPS-derived progenitors in the bioreactor system. We do hypothesize that to get a fully repopulated graft with the mature epithelium, we'll need to go for at least 21 or even 35 days of bioreactor culture in the presence of air. This, though, may require further in silico guided modeling and optimization with the Amon group in order to find conditions that would be better suited for mature epithelia. And so the idea then is to then implant these deepithelialized and repopulated grafts in orthotopic lung segment tracheal transplantation model. It's a fully circumferential model with Dr. Hakel, and really look to see optimize the point of transplantation. So do we want to transplant the day 10 grafts that have only seen air-liquid interface for 7 days, with the idea that perhaps the in vivo conditions would facilitate and increase the rate of differentiation, or do we need to put in grafts that need longer term differentiation in the bioreactor? And really, to be able to do these longer term studies in the porcines, we'll need to think about immune suppression regimens to be able to follow the IPS cells, or to move towards a non-immunogenic IPS cell line that we have collaborators for. So the other thing that I think we have a few more minutes, so I do want to spend a little bit of time showing you our other approach, which was to try to use a biomaterial in order to fully differentiate and create an epithelial sheet to them so that we could line the inner lumen of the trachea with. Here, this is work of Ratna Varma, who was a PhD student at the time, and she did an extensive search within the literature for existing biomaterials that could support epithelial cell attachment viability and differentiation. But for this particular application, we had to select materials that had specific criteria for mechanical properties as well. So they needed to be at a certain stiffness. They needed to be manipulated so that you could suture them. They needed to have a certain rate of bulk degradation. And in terms of the epithelial support, of course, we need to make sure that they had over 80% attachment. Their metabolic activity was healthy and maintained, that they could form these adhesions and they could differentiate to greater than 20% of ciliated cells and greater than 5% of goblet cells. And this percentage we selected based on sort of existing literature, suggesting sort of the minimum required epithelialization that could potentially lead to endogenous repair. So Ratna did a screen looking at all these different factors, and to summarize her work, she found that if we make a combination of silk fibroin that has really good structural support and mechanical strength, and collagen vitrogel membrane, which is really fantastic for differentiation and for growth of epithelium, then this composite could potentially be used to make these epithelial sheets. And in fact, found that the silk CV patches could support the both primary epithelial cells as well as the pluripotent cell sources. The cells formed a really nice epithelial barrier, shown here by the ZO1 expression. We could see that on the silk VM patches that had the progenitor cells on could generate ciliated cells, goblet cells, and there was no significant difference in terms of the differentiation capacity when we compare them to their matrigel in vitro control, which are generally how these cells are differentiated. And so just as a proof of concept model, we wanted to see how these cells would behave in the bioreactor system. Dr. Hakel made an airway defect model, which is essentially cutting up a little window in the tracheal wall and then removing the mucosa from that small segment. And then in lieu of the mucosa, attaching the epithelial patch that now has IPS-derived airway epithelial cells on top that have been fully differentiated, and then basically closing back this defect and running the bioreactor system for several days to see if the cells would survive, if they would maintain their differentiation capacity. And we found that they were fine, they were alive, they did maintain their differentiation capacity, their percentages of ciliate cells were approximately the same, percentages of goblet cells were approximately the same. So we're happy to move forward with these epithelial patches and test them out in vivo. And so we essentially, this model was created in vivo, and we established the timeline for epithelial repair. And so when you make this defect in the animal model, and then you cut it out, put it back in, there's an endogenous repair process that happens. So within seven days, there's a bit of granular tissue, there's no cilia, which starts to come back after 30 days. And by 60 days, you have an endogenously repaired epithelium where you had made your defect. And so this is the model that we wanted to use to evaluate the feasibility of using these IPS-derived cells. And while the ultimate goal is the long segment orthotopic transplant, using this model, we can get a lot of very valuable information that easily, without the need of sort of millions and millions of cells that we would need in the orthotopic model, as well as you can get a lot of information about how the cells will survive, what they will differentiate into, what happens to the grafts. And so we did a series of studies where we looked at sort of a control group where we just cut out the window, put it back, a stripped mucosa group where there was no cells and no silk, a bare silk graft where there was no cells, and then an IPS-derived SFCVM graft that was the sort of the experimental group that we wanted to test. And when we looked after 3 days, the animals survived. You could see in the control, the defect area is open. There's no sort of stenosis. In the strip mucosa, you can see that there's blockages starting to happen right here. And you can see a bit of that in the bare silk graft as well. And there's sort of narrowing of the airways within these two groups. And interestingly, we saw that when the cells were present on the silk CVM, this narrowing effect wasn't seen, and it was much less, if not any obstruction. And you can see that sort of better here in the histological sections, where you can see in the defect area in the strip mucosa, there's a sort of like a clear blockage that's there as well with the silk only, but it's not there in the when the cells are present. And although we don't see any pseudocytified no cilia, we do see pseudocytified epithelium. As I mentioned, the endogenous repair process is not within the timeline of when we did our experiments, and so we didn't expect to see the cilia. And then when we did look at the grafts, the cells were intact. We labeled the cells with a CMAC viable dye so that we can track them in vivo. And we found that after 3 days in the animals, cells were viable. The majority of the cells that were covering the graft were iPS-derived cells. You could sort of see very rare punctate single-cellular structures, but no ciliation, as I mentioned. And you could see sort of like that the goblet cells were there, and you could see a clear MUC5AC expression. And so really what we wanted to do is to see if we can evaluate these airway patches at a longer time point. These are human iPS cells in porcine models. So this initial study was just for survival and proof of concept. If we want to go longer time points, we again have to follow up with immunosuppression regimens, as well as perhaps using non-immunogenic cell types in order to derive the cells. The other thought, of course, is, although these epithelial patches can be used on their own in certain clinical settings, the idea would be, can we scale up these silk CVM grafts for use in the lung segment repair? So I do want to leave some time for questions. So the key takeaways from this work is that we've developed a rapid bioreactor-based partial decellularization method. This can remove the tracheal epithelium efficiently while maintaining the chondrocytes alive. And this leads to better structural stability for the bioengineered grafts that we can create. This reepithelialization is possible in a modified bioreactor system. So this double perfusion bioreactor system can be modified for long-term repopulation studies. We can even incorporate air-liquid interface culture within the bioreactor system for any amount of time. We've gone up to 14 days, but we can go certainly for up to 21 and 35 days to see if we can generate fully mature epithelium. And we've also shown that the iPS cells can be used as a clinically relevant source for repopulation of long segment tracheal grafts, as well as to use these silk CVM epithelial patches. And really we can gather a lot of necessary information from these in vivo studies that would help in our planning of the longer orthotopic transplant in vivo experiments. And that the iPS-derived epithelial patches remain intact after in vivo transplantation in the airway defect model. And this is really promising, because one thought was that as soon as the cells are in vivo, they're going to be eliminated completely, and they weren't, and they were maintained on the grafts. And so that's very positive. In terms of impact, as I mentioned, these silk CVM grafts are applicable for small airway defects, and you can adapt them for engineering any kind of epithelial grafts for other tubular organ systems. We can, and really the possibility of transplanting chimeric grafts that's generated from recipients' own iPS cells or immune evading engineered cells would prevent graft rejection and eventually circumvent the need for immune suppression. And that would be a huge advantage. So really this is a stepping stone for application into other systems, for example, the larynx, and it's we think is an advancement in the field of tracheal regeneration. So with that, I will end by thanking my research partners, Steve and Tom Waddell, and members of our lab, and the laboratory led by Christina Amon, David Ramiro, and Hank Yuli, who did a lot of this computational modeling work, and of course the Center for Regenerative Medicine in Boston, in particular Daryl Cotton and Finn Hawkins, for helping us to establish these directed differentiation assays in our lab. And I'm happy to take any questions.
Thank you so much. Oh, sorry, my camera is. Okay, there we go. Thank you so much, Dr. Karoubi, for the excellent presentation. I will open the floor for any questions. Feel free to put them in the chat as well. I'll give a few moments.
I have a question, actually, if I can go first. You were talking at one point about the shear test. Shear stress, is that similar to a friction wound?
It's so in a way, and so really we're measuring the amount of stress that the cells would feel as a result of the rotation in the graft. So our initial hypothesis prior to our collaboration with the Amon group was that perhaps the higher rates of rotation in a more dynamic environment would actually improve sort of cell distribution across the circumference and the length of the trachea. But in fact, it seemed that when we tested our lower shear conditions, the cells seemed to like the sort of more gentler approach and the less harsh conditions. The problem is that we need to have some rotation, because otherwise it would just sort of be the bottom of the trachea. And we've tried previously where we seed the trachea graft at a particular angle and then kind of rotate partially, seed again, rotate partially, seed again. But that results in again, not very homogeneous distribution of cells with sort of big gaps.
Okay, thank you.
Yes, Katie?
Hi, thanks for your talk. I thought it was interesting. My name is Katie Bain. I work at the CDTRP. I have a kind of, I don't know, it might be a silly question. Your bioreactor, how big is it? And like, was it scary to think about propping it up and maybe it like crashing down when you were like angling it, like in the lab?
So it wasn't too bad. I mean, it's kind of about this big. So let's say maybe 15 to 20 centimeters, and then there's sort of like areas within. The size of the grafts that we use are about 5 to 6 centimeters. They're not too heavy, and they've been redesigned to be quite easy to use, and so they're not really too heavy. And it was really a lot of graduate students and our postdocs finding out nice ways to sort of like hold it on an incline, and that, to be honest, was the best way for it to work. We tried several other complex ways to get the air liquid interface, and this was really the simplest method, was the best.
Oh, that's amazing. I used to work in research labs, and I know that sometimes if you like move things the wrong way or move equipment, and you could like void your service agreement and have catastrophe. So I'm glad that it worked for you.
Thank you. I think there's the question in the chat. Do you think your bioreactor could also be applied to the regeneration of the epithelial alveolar system? So I'm assuming that you mean the distal epithelium with the type 2 alveolar epithelial cells and type 1 alveolar epithelial cells. Okay, so if that's the case, no, and the reason I say no is that although we could test it out and potentially get some insight as to alveolar regeneration, in this setting, we do think that the geometry and the biophysical cues are really important in both the trachea and sort of the upper airways and in the distal epithelium. And the alveolar epithelial cells sort of reside in these hemispherical cell-like air, like grapes in the lung, that are undergoing cyclical stretch. So we do have other systems where essentially we're recreating an alveolus on a chip where the cells are seated onto relevant size hemispheres that are then pulled down using a vacuum to create this sort of cyclical stretch. And we do see that the cells respond really better compared to flat conditions. And the reason I think the bioreactor system for the trachea may not necessarily be appropriate is that there seems to be a threshold where the cell senses curvature. And we've done these studies before where if we put lung progenitor cells in tubes of different sized diameters, they essentially think they're in flat culture after a certain diameter, right? And so it's really important what geometry and what and the relevant size of that geometry they're exposed to. I'm not sure if that answers your question. And I think Patricia.
Yes, hi. Thanks very much for a really great talk. Thinking a few steps down the line with sort of your ultimate ambitions to get these types of grafts into patients, do you imagine, based on the physical properties of the tissue and perhaps the immune properties as well, that you would need to have a sort of a custom setup for each individual patient, or do you imagine that you would be able to not mass produce, but produce one tissue that would be able to be transplanted into any patient? Thinking about the size, like between pediatrics and adults, the physical size, right, but also other properties.
So that's a really good question. So in terms of pediatric versus adult, I think that would have to be significantly different, right? Like in terms of size. And so perhaps there'd be an approach for pediatric population and approach for sort of the adults. There are existing companies that are using porcine grafts as a potential therapeutic option, and they're really looking into sort of this pediatric size versus adult size difference that would be required. And it also it kind of depends on what the ultimate response in vivo is. So I guess two things could happen. One thing could be that this repopulated graft could serve as a bridge until endogenous repair happens, right? And so endogenous repair will happen. You will have sort of epithelium from both ends growing in. So depending on how long that takes. So if we know that based on the airway defect models that with a small window that, it takes up to 90 days for that area to be completely repaired. And so perhaps it would be much longer for a long segment tracheal grafts, but maybe once the reepithelialization process occurs, it's sort of faster along the way. So one thing is that they serve as a bridge to endogenous repair. And the other is that really you need to have a fully differentiated population of cells that will then, based on their in vivo environment, align, beat, and function as would native. And I think those we won't know yet until we do some of these in vivo experiments. Oh, to your other point, I know that there are some groups that were looking at bioprinting sort of very specific, sort of like patient-specific tracheas based on imaging, right? And so I think you can perhaps do that with bioprinted synthetic materials or a combination of bioprinted and sort of more naturally derived bioinks that you would use for printing. That's a potential option as well. But I think for these particular studies that we're doing in the porcine, the size is not really an issue, but it could be down the line, right?
If we don't have any other questions, I also have one. I'm curious what you would say your next research goals or research priorities, both for your project and for tracheal regeneration more broadly.
I think, we now know that perhaps fully decellularized grafts are not great. And we know that repopulating fully decellularized grafts are also not great. But there's been quite a lot of work done in the use of partial decellularization and perhaps the use of 3D bioprinting approaches with more naturally derived bioink. So I do think that the field is moving sort of towards using more better bioengineered grafts in general. In terms of for the field to move forward, we really don't know what happens in vivo, right? Even with these partially decellularized grafts, I'm sure they'll be recognized, right? They may not be rejected, but there will still be some sort of immune recognition. And I think as we learn more about the immune system and how it recognizes bioengineered tissues or grafts, we'll be able to sort of then reiterate our grafts and perhaps make them more enhanced so that they can be more non-immunogenic in vivo. But those studies really have to be done. And there's lots of new, there's a lot of interest, of course, in sort of these non-immune cells that are being genetically engineered with the idea of having a universal cell line that everybody can accept. And so I do think that there will be some work in looking at how those cells can be used therapeutically in tissue engineered grafts. For our own purposes, we really want to know how much information we can get in vivo using sort of these non-immunogenic, perhaps recipient-derived cells, and see what happens to them in vivo, see if we can use non-immunogenic cells, see what kind of information we can get in terms of the in vivo response to these long segment tracheal grafts.
Thank you. We have one last question in the chat from Katie regarding how many people a year get a tracheal transplant in Canada versus how many people are waiting.
Yeah, I'll be honest, I have to look up the stat to give you an accurate number, but tracheal transplants are rare. It's not something that is common, and I'm not sure how many wait for tracheal transplants, because I think you need to have a replacement right away. So I think the idea is to use a lot of these already existing replacement sources. It's just that they may need multiple surgeries, their quality of life is not great, and their grafts fail, needing further replacements, right? So it's more can we get a graft that will that can be used long-term in patients and give them quality of life and not have to be replaced or undergo several different surgeries.
That makes sense. Thank you. We're so used to hearing about solid organ transplant. It's not as often that we hear about non-solid organ transplant. So thank you so much.
Thank
and
you.
our time is just about up, but thank you for everyone's questions and comments throughout the presentation. And thank you, Dr. Karoubi, on behalf of the CDTRP for your presentation. If anyone has more questions, comments, or suggested topics and speakers, please contact Erica Croft at ekcroft@cdtrp.ca. And thanks everyone, and enjoy the rest of your day.